scrofa, which is pigs, which is a scientific name for pigs. So today we're going to be going over an update of what we've accomplished so far. Yes. So I want to start with an analogy. We all have pets, or we have people around us that have pets, and we all know that cats and dogs have different nutritional requirements, so we should buy tailored pet food for them. And this is because they have different amino acid needs. Cats require 11 essential amino acids, specifically taurine, which dogs can synthesize. And cats have a higher tolerance for vitamin D. If a dog consumes too much vitamin D, they can start having health problems. So every once in a while, if a dog nibbles cat food, it will be fine. But if too long exposure or depending on cat food will lead to health problems. And even though different dogs could eat different dog food formulations, we have a wide variety nowadays because the nutritional requirements of puppies and senior dogs differ, and the nutritional requirements of different dog breeds vary depending on their size, their age. The same is true for different cell cultures. It should be cell line specific. It should not only be tailored due to the organism that they originate from or their cell type. So an adipocyte does not have the same cell culture media requirements of a neuron or as a muscle cell. But even within the specific cell type, each cell line will have a different cell media requirement. And this is seen at the pharma level or the biomanufacturing level in CHO cell lines. Not all CHO cell lines are the same. There are different branches. Within these different branches or like large clusters or families, you have different cell lines and they all have different media requirements. So what does biomanufacturing pharma do nowadays? So apart from the sign of experiments and doing a wide array of tweaking to determine which specific metabolite or amino acid or transcription factor they need to tweak to increase their productivity, they are moving towards using genome scale models to drive the CHO cell media design optimization. So this was published in 2020 by Chow et al. This was done in Bristol Myers Squibb. So they used a genome scale model to basically predict modifying which amino acids, in this case leucine and valine, will lead to an improvement in their antibody production titers. And we'll go over this example a bit more in depth later. So what are genome scale models? Genome scale models are bridges that connect different omics data. They allow us to integrate different genomics, transcriptomics, proteomics, epigenomics, metabolomics data. But they actually are, what they list are all the genes and all the metabolites, all the reactions that happen inside a cell. And they list them out by having an index of all the genes and all their IDs. We can integrate all of these omics data. So for example, we can use transcriptomics data to constrain the different reactions and see which different cell types express which genes, and this can help you drive conclusions to understand a specific cell type better. I did a review, and I published a review paper last year, and I analyzed all the genome scale models that are out there for species that are of interest for the cultivated meat industry. And I realized that a lot of them only have partial genome scale models. And even if they have a partial genome scale model, they are not experimentally validated. Out of all this, an organism that caught my attention was pigs because they have a complete genome and all their metabolic pathways are elucidated, but they don't have a genome scale model and it has not been experimentally validated. So we thought that would be a good case scenario to show the cultivated meat industry that this is the future, and if we want to emulate the efficiency that pharma manufacturing companies have, this is what we should be doing. So that's what we're currently working on. And this is our project, the PigGEM. We are creating an experimentally validated genome scale model for pigs. And we aim to take this genome scale model and tailor it to at least 2 different cell types. We're working with myocytes and adipocytes so that eventually a company can use this cell type genome scale models to create cell line specific media.
Yeah, okay. So as Sandra mentioned earlier, these genome scale models contain all the information of the metabolism of a specific organism, specific species, or cell type, cell line. And how do we know what to put there? So you also mentioned that we can integrate different types of information in these models. It starts with the genome, and how do we know which chemical reactions an organism can carry out? That all depends on the proteins that it can fabricate inside its cells, more specifically enzymes that can catalyze all those conversions from one metabolite to the other. So once we know the genome sequence of the organism, we know which enzymes it will be able to produce to drive which reactions. And that is how we can use that information to build a model by making this network where we have metabolites as nodes and that they are linked by reactions. And those are directly associated with what we know is in the genome of the organism. And those are called gene-protein reaction relationships, and are one of the bases of these genome scale models. And once we have built this network, metabolic network, we can predict the rates of production and consumption of all these metabolites inside the cell, or what is the rate that they are being consumed from, for example, the cell culture medium, or that they are being excreted, and how changes in what is available to them in the medium will affect their metabolism. And then we not only have the information that we get from spent media, sorry, but we also know what's happening inside the cell by looking at what the model says.
Sorry about that. Yes. So another useful thing that we can do with genome scale models is flux balance analysis. So flux is the passage of a metabolite through a given reaction over time. And in genome scale models, we can create a stoichiometric matrix, which lists all the reactions and all the metabolites in a giant matrix and multiply this by a vector of fluxes of all the flow of these metabolites inside a cell. And we can use this matrix multiplication to achieve an objective. In the case of cultivated meat companies, the objectives that companies want to achieve is usually increasing biomass accumulation. So we set that as the objective function, and by solving this matrix multiplication, we can ask the model, so what is the ideal cell culture media composition that I need to achieve this objective? So the model optimizes all the using all the information that it has about the reactions and the genes in the cell. It will tell me what amino acid profile, what fatty acid profile, what glucose, what different media compositions we can tweak to achieve these objectives of biomass composition or growth rate, for example. This is what is done. The example that I mentioned earlier that is done in biomanufacturing. This is the Bristol-Myers Squibb example. What they did is that there is already an existing CHO genome scale model, which was published in 2016. And they customized it for the cell line by integrating transcriptomic data. And once this customized genome scale model was constructed, they create a hypothesis, and the model hypothesized that tweaking the cell concentration, the cell culture media concentration of 2 amino acids will drive amino acid production titers. So they were able to improve the productivity of an antibody that they were producing just by tweaking the concentration of leucine and valine. And the model originally predicted a 1.46-fold change. They were able to experimentally validate 1.33, but this is a really good improvement. And this is just one example. In another example, Victoria can further explain it.
Yes, this is actually a very new paper. It just came out maybe a week ago, but I thought it was especially interesting for to exemplify the utility of GEMs for cultivated meat. So what they are trying to do here is that they are using dimers in the medium also for CHO cells, also antibody producing CHO cells. They are trying to make a concentrated medium using a dipeptide with tyrosine. And they want to use the model to predict how this will affect the production of the antibody and whether there are other changes they can make to the media to improve their antibody production. And for that, they use the GEM. Next. And what happened here was that first they were able to predict what the best concentration of the dimer was, and then with the model suggested, let's say, or they made the hypothesis based on what they could see from the GEM, that now with the dimer they could also reduce the amount of asparagine in the medium and of glutamine, which makes sense because they were using a dimer with glutamine, and that this would improve all the production of the antibody. And they were able to achieve, by testing all these hypotheses, an increase of 9% in the production of antibody. So I think cultivated media might also be interested in something like that, in using concentrated media as cell densities go up, and also as perfusion is used in processes. And this is just another example of how GEMs could be used to assist the improvement of bioprocesses.
Yeah, so when you are creating a gem, we have this goal of being able to provide a starting gem that companies can eventually customize for the specific cell line. But we first need to start creating the baseline gem for the species. So that's what we're doing for pigs right now. And most genome scale model development follows very defined stages, and it has been true for human, for CHO, for rats, for E. coli, for yeast. You start by doing a lot of biomass composition studies and creating a draft genome scale model, which we already have. And with the biomass composition study, you create a biomass equation, which allows you to do flux balance analysis. And nowadays, we have a way of scoring or validating in silico how good a genome scale model is. And this is done with MeMode, and it gives you a score, and you keep iterating until you get a good model. And to further validate our genome scale model, we do experimental validation. So we do metabolic flux analysis to identify if the fluxes that the model predict are correct and the bottlenecks that the model predicts are the actual ones that we observe in the cell. And we also want to integrate transcriptomic data to be able to create or extract the cell type for at least adipocytes and myocytes, and also validate the model using spent media analysis to see if the uptakes or the consumption of cell culture media happens in the cells as the model predicts. So this is what we have accomplished so far in our project. For the wet lab portion, we have procured cell lines and isolated cell lines. We secure a collaboration with Colorado State University for Collins, and we were able to procure biopsies from them and isolate different cell lines from them. And we also acquire a satellite muscle cell from Oxygen. We characterized these cell lines, and I also started carrying out biomass composition studies for total protein, total starch, fatty acid profiles, and did some initial spent media analysis studies to identify if we needed higher volumes or higher concentrations in order to. I have a GCMS for doing the readings. I wanted to start initial spent media analysis to know if my signals were good. For the dry lab, Victoria can go more in depth.
Yeah, so for the dry lab or the modeling part, one of the first steps was to identify a template organism, so which ones are close to the pig and have a gem available. Select one of those gems. Also select a reconstruction tool to make our first draft models. And we created those drafts, and then we go to the evaluation with Mimode that runs some tests and tells us just some more about where we need to check our models. And then we, based on these scores, we selected two of the models of the draft models that we generated, and we went on to curating. So yeah, we will go more in depth on this.
Yes, so as I mentioned, we were able to procure biopsies from CSU for callings. These cells were isolated by carrying out the usual digestion to digest the extracellular matrix. They were incubated and strained and centrifuged to isolate the individual cells. And after culturing and passaging, we were able to get individual cells, and these were the ones that were used for the preliminary biomass studies. And we're also working with cell lines that we have acquired from Oxygen. So this is like the growth curves or some images from muscle satellite cells. The reasons we are working with both biopsies and cells that are acquired is that there is always the possibility that the metabolic profile of cells that developed inside a living organism and in vivo cells, over time they drift and their metabolism may look a bit different. So in order to have a better image or to be sure that the biomass equation that we're going to have in our main GM scale model is as applicable to a wide array of applications, we are going to average the biomass composition for multiple cell line origins. So we're going to be using adipocyte muscles, fibroblasts, both from biopsies and from in vivo cells that we have acquired from Oxygen. This is the biomass content from the cells obtained from the biopsies. So you see that their carbohydrate and total protein concentrations vary by cell type, and the protein concentration of myocytes is higher as expected. And I am currently working on the specific amino acid profiles and ratios for to identify which is the actual amino acid composition of these cell lines. So this is just preliminary GCMS data. I have also started preliminary spent media analysis, mostly just for glucose uptake and lactate excretion because the model needs it. So as the days pass, the cells are consuming glucose as expected and excreting lactate. And these rates are necessary as constraints in the model so that it is biological constraint in what the cells actually use.
Yes, okay. And then I will talk a little bit more about the modeling part and how all the experimental work that Sandra has been doing will tie together with the model. So as I said, first we wanted to find a template model or a reference model, reference organism for our project. So we look at the organisms that are more closely related to pigs. We have first cows. We also have goats, and then mice and humans. Out of these first four, there is metabolic reconstruction for cow, which cannot really be called a model because you cannot make simulations or predictions with it, but it's just kind of putting together all the metabolic information of cows. And then we also have gems for mouse and human. And from these, we decided to select the human gem as our model or template. Next.
Yes, sorry.
And yeah, we do this because this human gem just consolidates the knowledge of other gems that have existed for human in the past. So it puts a lot of this knowledge together in one gem, and it's also community curated. So it is continuously updated by its users just via an open source platform. And I think this is especially interesting for us as well, because that's kind of what we want for the PigGEM. We want to have this generic model that can be curated by the community, because everyone can benefit from it and then use it to make their own cell line specific models that they can apply for their own processes, or to optimize the medium for their own cell lines. Yeah, and another reason why we chose humans is because there is a good pool of information or data that compares humans and pigs, because pigs are used as model organism in research. And overall, the human metabolic network can be mapped to 94% of other, or 94% of the network can be mapped to other mammalian species. And more specifically, we have sources of data like this Pig RNA atlas that compares the genome of human and pig, and lists which genes are overlapping. And more specifically, it also has transcriptomics data available for different tissues in the pig. So it's also valuable information for our future cell line or cell type specific models. Next. And now we know we have a template, we have the metabolic network on KEGG, we have the sequence genome of pig, which is publicly available. And we selected this reconstruction tool called Raven that we use in MATLAB. And basically, you can use all these three inputs I mentioned on Raven to get some gem drafts. You could build it from zero, of course, but the idea of these tools is that they automate some of the steps. Yeah, and it also comes with other functionalities, but for now, I want to focus on the model reconstruction part. Next. So yeah, as I mentioned, we have all these inputs. We have the genome, the metabolic pathways, and the human template. And we use them with Raven to create our first drafts. And we created one draft based on the human gem and the genome sequence of pig. We created another one just based on the sequence and on the MetaSeq database. And we created a third one just using the KEGG metabolic pathway map for pigs. And all of these draft models, oh no.
Okay.
As you can see, all of these draft models are different. They have different number of reactions, metabolites, and genes. So just to select two of them, which is what we wanted to do, we go to me mode. We run some tests on each of these models, and me mode just runs a list of different checks and gives you a score for each of the tests. But we focused on the very first of them just because the other ones are less relevant when these models are just so preliminary or just a first reconstruction. And based on this, we chose the human-based draft and the KEGG-based draft for further curation. And the idea is that we will be curating them separately, to see if we arrive at the same point as a validation. And there are many different things to fix in these first drafts, and many different ways of fixing them. But just as an example of one of those things, and for the sake of time, I will go into one of them. One of them, this example specifically, is about molecular formulas. This is one of the first things that you can fix in these drafts. And also gives you an example of what you can see inside of them, or how they code all this information. So for each metabolite, you have the chemical composition or molecular formula. And this first, I identified that there were 108 that had an erroneous formula that just meant that it was not in the right format for the model. I pulled some information from the database INCHI to compare the molecular formulas I had in the draft with what's in the database. And then I corrected them based on what I found. And then just by doing this small tweak, I can improve the mass balance of the model, which is very important because we are talking about chemical reactions and predicting the rate of producing metabolites. So it's important that each reaction in the model is balanced, and the model uses all these molecular formulas to make those calculations. So now we went from 58% to 77%. And like that, by making little tweaks, different types of tweaks, we will be improving our PigGEM model. And some following steps will be gap filling. So this is kind of one of the main things that needs to be done for new reconstructions. Just make sure that the network is fully connected, that they're not missing links. And for this, we are going back to the human gem, just relying on the similarity between pigs and humans, comparing the get pathway map for pigs with the metabolic reconstruction of humans. And then we iterate, we reevaluate with me mode, and we keep pushing for a better model, more complete. So yeah, then as I said, some of the next steps are gap filling, make sure that our model is fully connected, that everything is in there. Then we will integrate the biomass reaction. So right now we cannot do any of the flux balance analysis, any of the predictions that Sandra mentioned, mostly because we don't have this biomass reaction, and that's where the experimental data will come in. We will build an equation based on all the measurements that Sandra has taken, and that will enable us to make predictions with flux balance analysis. And later, when we know that our model is consistent with also the metabolic flux analysis that Sandra will do, we can use it to make new media formulations, or just to create a protocol that cultivated meat companies or researchers can use to improve their media.
Yeah, so just as to recap our progress, we already have the preliminary biomass composition studies. This is a work in progress. I am still waiting for some cell lines that I will be receiving by the end of this month. So, and I'll just keep iterating to get a wide breadth of biomass composition studies. We already have the draft for a GM scale model. We can carry out flux balance analysis using the human biomass equation right now, but ideally we'll replace the human biomass equation with the pig biomass equation that that's why I'm doing the biomass composition studies. And we are working on the cyclical validation with me mode. And as I mentioned earlier, I've also started setting down the protocols for the spent media analysis that we'll be carrying out in the experimental validation phase. And all to drive this home, I really want to emphasize that our goal is to be able to create cell line specific gems, or allow people to create cell line specific gems in the future, so that we can be able to make the same predictions that pharma companies, biomanufacturing companies do, that they use a cell line specific gem to predict tweaking which components in the cell culture media will drive improvements in the process, be it for antibody production or for biomass, which is the objective in our case. So with that, thank you very much. We are open for questions. I don't know how we will be mediating the questions actually. But I see that Sebastian has his hand up. Yes.
Hi, Sandra. Hi, Victoria. First of all, congratulations for your talk. Very interesting. You have made a lot of progress, and I think this is the way to address this problem. So, congratulations for that. Uh, I was wondering a few things about the draft models. For example, you have three draft models, right? Uh, did you map the reactions in these three draft models just to see how much they overlap? Um, I guess it's kind of difficult because you don't have compartments on this
Yeah,
cake or metapsych, but you also have easy numbers or other things that you could map. So maybe you could see how much they overlap just to see how much you gain from each of these tools, right?
yeah, yeah. I have indeed done this, but just between the cake-based model and the human-based model. Um, if I am remembering correctly, it was something like 50% of the genes in the cake-based model are in the human-based model, and but these are of all genes, so that also includes some that were predicted through sequence alignment, so through BLAST, and then from the ones that were part of the metabolic network, it was something like 83%, if I remember correctly.
So
Perfect.
yes, we are indeed comparing because some of them are there's going to be an overlap, but right now we are choosing to curate them and then maybe eventually merge them together or choosing the one that is strongest or like has the best prediction.
Perfect. Perfect. And just one last question, maybe have you decide about which gap filling algorithm are you going to use? Because there are plenty of them.
Yeah, not really, not really. And I would love suggestions.
Yeah, I have some ideas, so feel free to discuss this later if you want.
Yeah, yeah, we can talk about it.
Yeah, we'll appreciate it. We use multiple ones here in the lab, but it will be suggestions are appreciated.
Perfect. Thanks a lot.
Yeah, for now, I was just eyeing out the one that comes with Raven, but I haven't necessarily compared any others that are available. Yeah. I see Simon. I think that's the
Ah,
only one that I see.
thank you.
Simon, yes.
Great, great presentation. I really appreciate it. All the pictures and going up to the higher level that I have a hope of understanding. So I have a high level question. You mentioned the gem can be applied to optimize media formulation to say maximize the biomass. And we know from the motivation behind the CMMC's scale down project with Merck was that they're finding that the media that was optimal in well plates performed poorly at larger scales, and they're trying to understand why. And so I'm wondering if the GEM model is used to optimize media at small scale, how does its application change when then optimizing the media for say biomass production at larger scale? How do those applications compare? How can it compensate?
Um, so it is known that like the bioreactor that you use to culture a cell affects the metabolism. There has been studies that you can see that the fluxes change. This was done with duck cells with the initial duck genome scale model reconstruction. They did metabolic flux analysis and they did it in different bioreactor types, and they found that culturing the cells in a single layer was different than using CSTR but than using like a hollow fiber reactor. So the cell's metabolism change. This is something that my PI has brought up. So eventually in the metabolic flux analysis phase during the experimental validation, she does want me to carry out experiments both at the plate level, and we have 5-liter bioreactor available. So I will do some of the metabolic flux analysis studies in the bioreactor, and this should give us an idea of how experimentally we see a change in metabolism versus a 2D format and a bioreactor format. The answer is that once we see it experimentally, we could integrate that knowledge into the model. But since we do not have like a way of carrying out the flux balances analysis right now, I do not know how that would look like. But yes, there is a difference. It does, like the bioreactor, the environment affects the metabolism of cells, and we could eventually integrate this into the model.
So it would be the same unified model, or would it be a different application? I mean, would it be a different, yeah, or would you need different models?
It is possible that in the different environments, the constraints that we see in the cells are different. So we would just need to adjust the model so that we can constrain the model in the same way that we see. For example, let's say that glycolysis works differently in a bioreactor versus in a 2D tissue culture. We will constrain, since we're seeing that experimentally, we will constrain that in the model so that, or we will tell people that to constrain that in the model between the two different growth conditions. That's the way that I envision it doing it. But the correct answer, we don't know yet, but we have considered that as a possibility for the experimental phase.
We have some.
Thank you, Sandra and Victoria, for a wonderful presentation. It looks like we have a couple more questions in the chat as well.
Okay.
So I can read it to you guys and yeah, see if you can give a response. So Kevin said, thanks for the presentation. Is an organism level GEM a prerequisite before a cell type / cell line specific GEM can be constructed? And if so, would the biomass composition studies be different?
For unicellular organisms, you don't necessarily need to build an organism level GEM. So if you can bypass that, if you have the sequence for a specific cell type and you carry out all the biomass composition studies for that specific cell line, you could bypass the organism level GEM. But in this, and yes, that's like a question that we've had. Should we have the different biomass equations depending on the cell type? So far, what we've settled on is on having a unified biomass equation like the CHO gem has, or like the human gem has. But the plan is to still make available the biomass composition studies results that I will have for muscle cells, for adipocytes, or fibroblasts, which are the main cell types that I'll be working with, so that if we want to test if changing the different biomass composition equations depending on the studies actually affects the model performance. So we'll be doing two approaches. We'll be keeping the biomass composition per cell type and also creating like an average, a weighted average for the general one. That's the approach that we have settled on so far. But we'll see. This could change because after we start experimental validation, we'll iterate and see if this is the best approach. It may not be. We don't know yet.
Okay, great, thank you. Kind of a great follow-up question too. Agassiz asks, hi, thanks for the presentation. I was wondering what kind of cells you were going to use for experimental validation, and also what assays are you going to run?
Yes, so the two main cell types that we thought will be relevant to begin with will be myocytes, skeletal muscle cells, and adipocytes, because we think those are of interest for the cultivated meat industry. I also have fibroblasts as a comparison, as an extra cell line, but the main two ones are going to be adipocytes and and myocytes. What assays are we going to run? So for spent media analysis, it'll mostly be, apart from like glucose uptake, lactate uptake, also see how the different amino acids change on a day-to-day basis. So how to see how the cells are uptaking it, but also what seems to have been done by other groups, so by other research groups, is that they just do ELISAs to see how the cells are uptaking certain vitamins or certain transcription factors. So we thought that that would be the way to go. We routinely carry out ELISAs in the lab, so it will mostly be using GCMS, gas chromatography, mass spectrometry for everything that is amino acid uptakes, a YSI or a CDX bioanalyzer for glucose uptake and lactate production, excretion, and for everything else, it will be ELISA assays. I hope that answers the question.
Yeah, and maybe also one that is important is the metabolic flux analysis. That's also
Yes,
part
so
of
yes,
the validation.
Yes, so to validate the model and to see that the flux balance analysis predictions are correct, I'll be doing metabolic flux analysis. And to do that, I'll be feeding the cells a labeled media labeled with a radioisotope. So cells will consume it. After some time, I'll analyze the cells with GCMS. So they'll tell me which metabolites were tagged with this radioisotope, and I'll be able to see how the metabolites run through the system. I'll be able to create a flux map, and this flux map will be compared with the flux map that we get from the flux balance analysis prediction. So that's another important part.
Great, thank you. I think Elliot has his hand raised next.
Hi,
Nice
Elliot.
job, Sandra and Victoria. Hello. So I think what I'm interested in knowing is, this is the cultivated meat modeling consortium. And as one of the first sort of projects that's funded by the consortium, I'm curious how might you envision like CMMC members or other people in the community being able to collaborate on this project or other sorts of projects that are like it? Like what strikes me is, part of the reason why we need these sort of consortium and collaborative approaches in this industry in particular is because everyone that wants to do media development or cell line development, I mean, everyone has to start from scratch and do the same thing. And so by having people come together and collaborate, you can sort of build off of work that's already been doing. And, for instance, people can maybe share omics data that can plug into this, or people can share biomass composition data that they might be, taking in their own labs from their own cell lines and cell types. So I'm curious if you have any ideas of ways the community could get involved in this project, either as a springboard to collaborations, or even starting similar projects that are related to this in different species. Have you been receiving interest from people within the CMMC or outside of the CMMC? Has that led to any ideas? I'm just curious.
Yeah. I think I wanted to go back to when I mentioned the human gem and how it's community curated. I think that's a good example of how I would like the PigGEM to help, let's say, the community and to engage people. It won't be perfect when it comes out. I know that for sure. Like the human model gets updated and gets just tweaked more and more, just to make it a bit more accurate or just better for its purpose. So I would like that as people, I don't know, use it and maybe compare with what they are seeing in their cells, that they can also help us make certain changes that they suggest them. At the same time, I think what you mentioned about sharing omics data is ideal. And for us at this stage, I think it's actually very important. So if there's anyone here who has some big metabolomics or transcriptomics, it's really helpful. So yeah, that is definitely one good way that we could get engaged. I think there are also people in the CMMC who do more and maybe machine learning or these types of things, that's also important when analyzing maybe the this data or the outcomes of the model, or maybe integrating models with something else. Yeah, those are the things that now come to mind. Maybe Sandra, you have more.
Well, we have had interest from different companies because ideally there's I would say the challenge in the cultivated meat industry is that so many people are working in so many different organisms, and it means that multiple models will be needed eventually, and different companies have approached us for helping maybe eventually create these models. Something that I see as a constraint so far is isolating the cells or getting access to the cells, because we need the biomass composition studies, and to eventually validate everything, we need metabolic flux analysis. There is definitely potential for a lot of collaboration, as Victoria mentioned. Ideally, we will be able to have a database like some of the human model, the yeast model, the CHO model. They are all housed in databases like Big. Keg houses some of them, but like I can actually download the publicly available model in Big. So that would be ideal. We could create like a database for all of the different organisms, and people could use it as a starting point. And this actually ties back to one of the questions that I had in the chat as to why is an organism level a prerequisite before a cell type. Once the organism level is created, if multiple people are working with pixels, they don't need to start from scratch. They can just like take what we will be publishing as a springboard, as a starting point, and they would just need to tweak it a bit for its own specific cell lines. I think it is it's a big effort for a small company to dive in to create a huge organism level model. If we already have like different starting models for different organisms, people can easily customize them. But yes, it does have the potential to, I'm sorry, it does have the potential for collaboration, a lot of collaboration in the future. The CMMC could definitely become a meeting point for people to discuss how this could work. Different people have suggested, I don't know, sharing dried cells. I know that cells are a very important, a touchy subject in the cultivated meat industry, but even if I don't get live cells, just having dried cells for biomass composition studies will be helpful for starting the big part of the experimental section, for example. But yeah, I don't know. I'm sorry,
Yeah,
I'm
and
rambling.
I'll just add, like, I mean, I think people here should know, like people on the call, that the CMMC is supposed to be a point where people can come together and talk about these sorts of things, because the community is so small. There's not a lot of people doing this. It could, it's a way to springboard new ideas, and also meet new people that, you might, collaborate on a new grant proposal, for instance, and build off of this existing work. So I think that's would be a great outcome to build on this project, is to get people interested, involved, and figure out how to progress it as a community effort. So thanks.
And now that you mentioned collaboration, something that we did mention in our proposal is that I am establishing the protocols for like all of these biomass composition studies and metabolic flux analysis, and they are sort of general, but sometimes you need to tweak a bit when you're working with yeast, or when you're working with algae, when you're working with mammalian cells. And I've already tweaked them for pigs, so I can easily share. We plan on sharing some of these protocols so that people can replicate this if they want to. It makes the model stronger. The community makes the model stronger.
Also
Yes,
something.
Alex also has a question.
Maybe I wanted to add something small before the question. I think also you mentioned other species. I mentioned earlier that pigs and cows are very closely related. So hopefully we expect that when our PigGEM is fully curated, it just is a good starting point, just like we use the human gem as a template that the PigGEM can be used for these other related organisms.
Yep. Alex has a question. Hi.
Yeah, thank you. It looks like we have time for a couple more questions. Yeah, you see Alex has his hand raised, and then we have a couple more in the chat. So if Alex wants to unmute. Okay, looks like we're having some trouble with the audio, so maybe Alex, you could add your question to the chat and I can read it for you instead. But let's go on to the next one. Rickard asked, what does an ideal omics data set look like in the context of GEMS? If you weren't funding/time constrained, what would this look like? Would love to hear a bit more about how these different data sets within a given organism can be integrated with one another as well. Thanks. And then, sorry, a quick follow-up too is also would love to hear more about industry interest in the work. Are you guys currently collaborating or in talks with companies? How has the reception been in industry?
Does
Um,
do you
well,
want to go?
so yes, so ideally, we would like to make the model available when we publish it. This could be on GitHub. The ideal omics that we will like start to work with will be transcriptomics, because there are good packages out there like eFlux that allows you to use the transcriptomic data to constrain the model and customize it for your specific cell type. So just like out there, the Pig RNA Atlas or NCBI Geo already has dataset for pigs. These are datasets for pigs that are used in clinical studies. Ideally, we could have similar datasets for pigs in a cultivated meat context, because a lot of the time when we are curating some of these datasets, we need to make sure that the pig hasn't been treated with any drugs that they're testing, or that they are not genetically modified animals, for example. So having transcriptomic data sets for animals, for cells that will eventually reach the consumer will be ideal, and a good point to start will be transcriptomics. Yes, and as I mentioned, we plan on making the model available, maybe in a GitHub repository. Maybe ideally we will be added to big. That's dreaming big, but yes. I don't know if you want to add anything, Victoria.
Yeah, I wanted to say, so you talked about transcriptomics, there's also proteomics. Both of them can be used, for example, to go from the generic model to go to the tissue-specific model. And at the same time, we have metabolomics, which is more maybe for cultivated meat for the media design. So if you know what is inside the cell also with the metabolomics, and then outside the cell with the metabolomics, and then you can link it with the information that you're seeing in the model. That can, so usually metabolomics just tells you what has come up, what has gone down, and then if you look at the model, you can have a better picture of the mechanism through which everything is happening. So that's also another way of tying this data to the model. Mostly, yeah, proteomics and transcriptomics are used to make these more specific models. So the idea would be that if a company has the transcriptomics and proteomics of their cell line, then this can be used to make their specific cell line specific model.
Yes, I see the follow-up from Alex in the chat. He's from extracellular. Yes, we have the CMC has talked about extracellular, and that you guys want to get involved. We definitely would like to jump on that opportunity. So we should set up a call, because so far one of the big constraints that we've had was setting the collaboration to be able to source the cells. that took us a long time, and isolating the cells is also a bottleneck for me because establishing a cell line is not trivial, and then I need to, once it's established, I need to start the biomass composition studies and everything else that we'll do for experimental validation. So yes, I would say we should set up a call.
Sorry, Sandra, jumping in. Alex, thank you so much. I think we've emailed Nico. I will send an email again to connect Sandra to you guys again.
All right, thank you, Sruthi, and thanks, Sandra. There was one more follow-up from Sebastian, and I think you already addressed about sharing resources on GitHub, but he also asked how much time you have left for this project. And yes, as you answer that, I will share one slide too to advertise our CMMC crowdfunding campaign as well. So thank you.
Yeah, about I will mention GitHub again. I currently am using it myself, just with the integration that Mimode has, but probably if it's to come out to the public, maybe that's also a way in which we can engage the community, because I'm definitely not an expert in hosting something. So, oh, and in terms of how much time we have left, so this is the midpoint review. So
yeah,
we are sort of halfway. But yeah, but hopefully there will be more funding to keep it going. Maybe from Sandra's side.
yeah. So to finish this initial thing, we have around 6-7 months to go, at least on the experimental side. And this will be just for the initial model. My PI does have a lot of questions at using this as a starting point, because cell metabolism changes with differentiation. There's a metabolic shift, something that we would like to explore. We would like to be able to see or explore more how the different bioreactor, like the 2D versus the actual bioreactor environment, affect the metabolism. So, but to do all of that, we are applying for more funding, but we're using this as a starting point or as a springboard for more. So yeah, I don't know if that answers the question.
No, I think that was perfect. And yeah, no, thank you both, Sandra and Victoria, for a wonderful presentation. And thank you everyone for attending. Really appreciate the time and support that you all have given to this project. And yeah, definitely excited to see the next half and future advancements as well.
Yeah, feel free to reach out, I don't know, through the CMMC, through LinkedIn. Yeah, thanks for all the questions.
Thank you.
Excellent, thank you. And yeah, we'll be adding the recording as well, so you can have a good reference. Okay, but yeah, thank you all, and hope you have a nice rest of your day.
Thank you.
Bye.
Thank you. Bye.